have a elaborate plan in place that Cody just detailed to me, so it involves costumes and dancing, so we're looking forward to that.
Everybody.
Oh, man.
Okay, and so there'll be a lot of people going in and out today for the Mount David Summit, and I will try to wrap things up in a couple of hours so that everybody can take advantage of it. And I hope that you all take as many opportunities to do research as you can while you're here and in the future. It's a good way to spend your life. Okay, answers to questions. First, then transplantation, and then immunization and HIV. I will go relatively quickly because we've had discussions on both of these topics. If I'm going too fast, let me know. Feel free to ask questions. Okay, so answers to questions from the last couple of classes. Why does smallpox vaccine leave a scar? So my answer was basically right. The smallpox vaccine is not given with a hypodermic needle. It's not a shot like many vaccinations. It's given using a bifurcated needle that's dipped into the vaccine solution. When removed, the needle retains a droplet of the vaccine. The needle is used to prick the skin a number of times in a few seconds. So basically, they make mincemeat on your arm with the vaccine, and then that results in a scab which falls off, and then you get a big blister. And so that the scar that you get from smallpox that some of the older of us have is from that blister from the active lytic viral infection. Okay, can Jehovah's Witnesses receive a solid organ transplant? We were talking about transplantation, and the specific issue is blood. So the witnesses view Old Testament prohibition against eating of blood as meaning that blood from one person should never enter another's body, even through medical needle. Blood transport bans, so they cannot donate or receive organs. However, as with any faith, there are less observant members who believe that you can wash organs for transplantation, thus eliminating the blood issue. So the blood is the issue, and depending on how observant you are is whether organ transport constitutes blood transport or not. Is there an insulin pump that, is there an insulin pump that tests blood glucose? There is. It's interesting because it looks like most insulin pumps are married to a separate device for testing, and but there are a number of different pumps. This actually wasn't the one I was looking for, but there are a number of pumps by companies like Medtronic that have automated glucose sampling. So if you just have a pump that looks like this, if you have automated glucose sampling and the pump that looks like this, and you can manage and track your glucose levels on your computer. So I know everybody was holding their breath about that one. Colin? Do you remember?
Yes, and I forgot.
Okay. Okay, we'll be expecting an answer next week.
Is it about the transgender?
Uh-huh.
Yeah. Okay, I could do that.
Okay.
Found a study that actually showed a male to female transgender man, or woman, sorry, developed SLE, and they thought it might be linked to the hormones.
There we go. She saved your bacon.
Can I eat it?
Okay, good. All right, we'll expect more next week. At the end of one, a case report is not a very convincing study. We need a placebo-controlled crossover, so to speak. Okay, so organ transplantation. We had a discussion a few weeks ago, and as we started discussing, major issue with organ transplantation is T-cell immunity, and that's because T-cell immunity is mediated through the interaction of the TCR with MHC and with peptide in MHC. And obviously, in unmatched donors, the T-cells of the recipient of a solid organ have never seen many of the HLA genes of the donor. So the problem basically is that transplantation defeats thymic selection. So when you have normal thymic negative selection, you have T cells that see self-MHC and peptide too strongly, and they are eliminated. However, there is all that self-MHC from the donor organ has not been seen in the thymus, and therefore it defeats negative selection. So there are two alloreactions that are happening in transplantation. You can have the self-MHC with foreign peptide, and you can have the foreign MHC with foreign or self-peptide. So there are two ways that T cells can see new antigens, either because of the change of HLA or because of the change of other proteins. Okay, and which type of alloreaction occurs depends on the source of the antigen presenting cell. So if you have a donor antigen presenting cell presenting antigen in the context of its class 2, then that's direct allo recognition. So it says, I've never seen this HLA before, and so it's foreign, and therefore I'm going to form an immune reaction against that antigen. And then there's indirect, where some cells or dead tissue from the transplanted organ are taken up by self-antigen presenting cells, and the T cell says, well, the HLA is okay, but this combination of peptide and HLA, that's not self. So that must be a foreign antigen presented in the context of self. So I have to form an immune reaction. Okay. So HLA is most important for transplantation, as the discussion group pointed out, but allelic variation of other genes produce minor histocompatibility antigens. So in the polymorphic proteins that are different between two individuals and get presented on MHC class 1, so any of the tissues of the self, in the original, if you were a kidney transplant recipient, your normal kidney proteins would be shown on your HLA. If you receive a new kidney transplant, even if the HLA genes are perfectly matched, some of the proteins are going to be genetically different in the recipient, and therefore the combination of HLA with peptide will be new ones that could potentially provide a basis for immune response.
Can it work the other way around then, if you have enough matching that even though HLA isn't matched? Is that a possibility for no immune response to be seen?
No, if you have HLA that is not matched,
Mhm.
then that's the basis for the strongest immune response, because you have not eliminated the T cells that strongly
Mhm.
bind that non-self HLA. So remember that in the thymic education, positive selection is you get enough activation through TCR to keep the T cell alive, but if you have too much activation, then they're eliminated. So most of the T cells that see just self HLA are eliminated, but there are a lot that remain that would see any other HLA. Okay, so, yeah.
So, in the case of transplantation and you say like they match the HLAs, but why do they still take anti-rejection medicine? So
Because of the minor histocompatibility antigens.
okay, so it
Because
basically
let's
does
say
this.
even if you are totally HLA matched, then you're not genetically identical to your donor, which means there's some genetic variation in proteins. So that's what this is displaying here.
Okay.
Even if all the HLA is the same, there are some other proteins that differ between the two of us.
So
And
that's-
those are going to be presented, and those would be like you were infected with a foreign virus, so you're making non-self protein and presenting it on self in HC, right? Does that make sense? Okay. So presentation of non-self proteins on class 1, so that's the CD8 immune response. So this can also happen, minor histocompatibility antigens can also be presented to CD4 cells. And this happens basically when the donor cells that have class 2, so the situation we just showed was where a donor cell with perfectly matched class 1 has the minor histocompatibility peptide presented to CD8 positive T cells. We can also have donor cells die and shed their proteins, and those proteins be digested by self-antigen presenting cells and present those minor histocompatibility antigens. So, Jenny, in either case, the T cell thinks what it sees is an infection, because this is exactly how an infection works. We have matched HLA with a new foreign peptide. In the case of a virus, it's going on class 1. In the case of an extracellular pathogen, it's going on to class 2. So even with a perfect HLA match, the minor histocompatibility antigens can lead to rejection because, as far as the immune system is concerned, what it sees is an infection. Does that make sense? Okay. So the result of either major mismatches in major histocompatibility antigen, HLA, or minor, the same process can result. So donor antigen presenting cells migrate to a local lymph node and stimulate alloreactive recipient T cells. And recipient, so they can take up foreign proteins from the donated tissue. Recipient antigen presenting cells can also present their own self-antigen. And in either case, you get recognition of of the transplanted antigens as foreign. And this basically recapitulates with the same idea that organ rejection is just like a process of infection, where in this case, the skin graft, you get migration of antigen-presenting cells, just like when you have a trauma or a skin infection. The antigen-presenting cells have taken up bacteria, and they migrate to the lymph node where they present antigen, come in contact with the specific T cells and B cells, and then effector cells migrate back to deal with the local infection. The exact same process occurs with the organ, where the antigen-presenting cells migrate to the lymph node, get matched up with the antigen-specific lymphocytes, and the effector cells go back to the site of what it thinks is infection to reject the tissue. So the more trauma or tissue damage that occurs during transplantation, the more activation of the innate immune response and the more likelihood that this will take place. And just like every other adaptive immune response, graft rejection results in memory. So there's a specific memory of the mismatched antigens, resulting in formation of graft-specific T cells and antibody responses. So in terms of in the clinic, when people receive transplants, any rejection of tissue can be characterized by the speed with which it happens. So there's hyperacute graft rejection, in which case there's already preformed circulating antibodies against the transplanted tissue. And so in very short order, the circulating antibodies get into the blood circulation of the transplanted tissue, see their antigen, form immune complexes, so you get immune complex deposition and complement activation, vascular damage, so that's the most rapid. Acute recognition is really what we've been talking about so far in terms of there's some preformed alloreactive CD8 positive T cells that see their antigen and then produce extracellular self-killing. And then chronic rejection is that process of taking up foreign antigens, antigen-presenting cells migrating to the lymph node and sending adaptive immune cells back. So it's really about whether there are preformed adaptive immune memory available at the time of organ transplantation.
Under what circumstances would hyperacute happen?
Previous transplantation would be the most likely case, or a case where there's been or a postpartum woman that's had some immune exposure to a mismatch. Yes.
So, I mean, I was just thinking, if it was for an older transplant, wouldn't it be different HLAs if you're getting another transplant? But if they were trying to match the HLA, I guess, yeah. Okay.
Yeah. So so hyperacute rejection is, I don't think it should ever happen anymore in modern transplantation medicine. I think it's a description of a phenomenon that when people were just randomly trying to transplant tissues, it's a description of how fast the rejection took place. But with current immunophenotyping, it shouldn't ever happen that an organ's being transplanted into when there's pre-existing immunity.
But if you were trying again, you would definitely try for like a different HLA set then?
Well, if you're trying to match self as close as possible, it's not going to be that different.
Yeah, you said, yeah, I guess you're in a whole
I
other
imagine,
host of
so
problems.
for each organ that gets transplanted, there's a whole criteria in terms of how matched they have to be and what the basis of the previous, if you previously rejected an organ transplant immunologically, then that patient is going to be is not going to be a good candidate for a repeat transplantation. Did you have a question, Ian?
Yeah. What happens, like, if I'm correct, if you get like the wrong type of blood transfuse, you die, or most likely die? Is that true?
Well, I guess if you had an anaphylactic response, you could, yeah.
So how would that occur? If you, this is all through the adaptive immune system. So if you don't have like a hyperacute reaction, then how would that be particularly damaging to you?
So red blood, so if you look in your book, there's a section about blood transfusions and what we all know as our blood types. Those are specific antigens on red blood cells. So the compatibility for blood transfusion depends on those red blood cells. And that is a case where you get acute rejection based on those. It's not based on HLA, it's based on antigens on blood cells. Because if you're type O or type A or type B, you have preformed immunity against the other.
Oh, okay. Oh, right. Because like, aren't those in themselves non-protein, like some like
Yeah, they're
sugar derivative
yeah.
or something?
Yep.
Oh, okay. So innate immune system actually does have recognition of your non-self blood type.
It's actually adaptive immune
Oh,
recognition.
okay.
Okay, so basically a transplant rejection is just like any other adaptive immune response where all the cells that we've talked about so far this semester get involved, be it the dendritic cell presenting antigen, helper T cells that recognize either non-self HLA or non-self protein in the context of self HLA, producing cytokines that lead to B cell maturation. So again, you can get expansion of antibodies against non-self proteins, and maturation of cytotoxic CD8 cells, and downstream activation of macrophages, NK cells, etc. So there's nothing different about the adaptive immune response to organ transplantation than against any other non-self entity. So we discussed a little bit during the discussion about graft versus host disease. So what we've been talking about so far is rejection of the transplanted tissue by the self-immune system. But if you transplant a sufficient number of lymphocytes, the opposite can occur. The lymphocytes that have been transplanted in the tissue see host tissue as foreign and produce an expansion and a response against the host tissue. So it can happen in both cases, and particularly in the area of leukemia and lymphoma, this is taken advantage of such that when people get a hematopoietic stem cell transplant, in addition to allowing chemotherapy to kill off the leukemia and the lymphoma, there's also an advantage to the graft versus host disease, where there's actually a graft versus leukemia or graft versus lymphoma response. So a slight amount of graft versus host disease in the case of blood cell cancers is actually advantageous, because it helps to clear the immune cell tumor. Okay, so how do we successfully do graft rejection? Obviously, we want to match HLA as closely as possible. Traditionally, this was done with laboratory tests of complement-dependent killing, complement-dependent cytotoxicity. Now it's done with a lot of molecular biology techniques. There's also testing of viral antibody that exist between the donor and recipient. And immunosuppression, to some extent or another, is always involved. And then induced tolerance is something that's still in the research phase, but we can talk about briefly. So complement-dependent cytotoxicity is basically the result of killing of cells by antibodies binding cells and activating complement. So what can be done in the laboratory is basically mix the sera, for example, from a recipient patient with blood cells from the donor patient, and looking to see if any of them are bound by those antibodies and activate complement. If they don't bind the cells and the cells aren't killed by complement-dependent cross-linking, then there are not antibodies to the HLA type. This was a traditional method for matching donors, however, and that's how MHC in humans came to be called HLA, because these were the human antigens that were used to match donors. So it basically looks like you have live cells in culture, you add recipient serum and complement, and if there's a mismatch, the cells are lysed, basically. They're all popped in culture. So then the next generation was to do molecular biology, and in previous decades this was done by doing restriction length polymorphisms, by digesting DNA with bacterial site-specific exonucleases and comparing the banding patterns. So the sequence was assumed by the digestion pattern. Now, most recently, they use PCR to amplify specific regions, and now sequencing is so rapid and and cheap that most people get completely sequenced in their HLA region when they're getting transplanted. There are also techniques for determining alloantibodies using flow cytometry. So basically, there are a number of beads that are engineered to express different HLA proteins on the surface, and they're mixed with sera from a recipient patient, and they shift to the right when antibodies are found and detected with a secondary reagent. So, the key is basically matching HLA and checking for HLA-specific antibodies. So this essentially is a microcosm of the interaction between an antigen-presenting cell and a T cell, and it shows all those different signals we've been talking about. So signal 1 being MHC peptide interacting with CD3, signal 2 being the interaction of co-stimulatory molecules, signal 3 being the secretion of IL-2 by the T cell and binding back. And so it then puts on that background all of the currently approved immunosuppressive treatments that are used in transplantation. So anti-CD3 monoclonal antibodies is a very potent immunosuppressive. Why might you not want to use that as an immunosuppressive reagent?
Would it cut off other signaling pathways as well, or stop other signaling pathways?
Well, it would stop every T cell signaling pathway. So essentially, it would be the equivalent of HIV. You would have no T cell mediated immunity at all in both CD4 or CD8 cells. CTLA-4 is actually a pretty effective biologic. So this is a soluble form of one of the coactivating molecules that blocks signal 2. There's also blockade of CD40 ligand interaction, as well as anti-CD25, which is the receptor for IL-2. There are a lot of inhibitors of the mTOR pathway, which is downstream signaling from cytokine receptors, and so on and so forth. But basically, everything that takes place for an antigen-presenting cell to activate a T cell can be targeted with the drug in order to promote tolerance to a mismatched graft, and what the doses you use and how many of them use in combination basically is customized for different organ transplantations and modified over time to have a balance between immunosuppression and graft rejection.
You have to do this constantly, like for the rest of their lives?
That is my understanding, but I'm not an expert in transplantation.
Does this overall make them more vulnerable to other pathogens or?
Absolutely. Absolutely increased risk of infections, absolutely greater chance of death by cancer after long term after transplantation.
Would it be possible for people's like immunosuppressive treatments to change over their life, or is it like you get treated once and then you just get monitored?
So, like I said to Caleb, most people have some sort of maintenance therapy throughout their lives.
But is it the same, or
It
is
doesn't
it?
necessarily have to be
Okay.
so. I think probably the dose in the cocktail is customized over time because the other thing is a lot of these treatments will have other side effects other than immunosuppression, right? An individual patient will or won't tolerate those side effects as well. I mean, they're minor, some of them are minor chemotherapeutic agents, so. Okay. So, knowing all that we know now about immunology, you might think that you could do some tricks to induce tolerance. And one way to do that is by depleting T cells. So we said that most of the graft protection is mediated by T cells, especially in the case of an HLA mismatch. And so if one treats, if one does a transplant into a mouse from another mouse that is not matched for MHC, the animal rejects the graft very quickly. However, if you deplete T cells from that animal, just CD4 T cells from that animal before transplantation, the graft survives longer. If you inject the CD4 antibody at the same time as the graft transplantation, you can then induce tolerance to the animal because there are essentially no T cells present at the time of engraftment. And so it's theoretically possible to tolerize recipients to grafts by eliminating T cells. So some kind of T cell conditioning is part of every transplant. Yep.
Similar to the treatment of some autoimmune diseases, is it possible to eliminate the T cells and replace them with the stem cells? And that will increase tolerance too?
Yep.
For
The
transplant.
problem there is that the transplanted stem cells could see all of the recipient as non-self, right? Although they would be getting, if the recipient had some thymic tissue left, they could be getting educated in the host, in which case it would work better. Ian?
As the, if you're eliminating all T cells and then you add the graft, is it still educated aren't the new ones still educated in the thymus and that would not present the graft genome? So, I mean, how does that, how does that, the method you described, get around that?
It's not entirely clear how that works. But it's thought that there are some mechanisms of peripheral tolerance that help. If you eliminate all T cells simultaneously with transplantation, then there's some peripheral tolerization of the new T cells that develop after that.
So like what if they're newly
They're all
proliferated, then they have a greater tolerance to what's okay.
Okay, so the future is something akin to what Colin was just saying. If you're transplanting a solid tissue, then you can transplant hematopoietic stem cells from the same donor, so then they're HLA matched. However, as I pointed out, the donated immune system and the host would then be mismatched. So you would need to transplant some mixture of thymus such that the new T cells that are educated both to the donor and the recipient thymus. So they are tolerant of all the recipient HLA genes as well as the donor HLA genes, so that you tolerize the new T cells to both sets of genes provided by the donor and the recipient.
If the donor and recipient, if they are like related, like parents or like siblings, do they still need to do this? I know like their combination is very unique, but like is like less proportion.
So there's whole huge sets of flowcharts and rules about matching. It's mostly about how much the HLA can be mismatched, and then how much the minor compatibility can be mismatched. So currently, they try to match people as closely as possible. Obviously, the likelihood that you are genetically closer to a family member than an unrelated donor is increased. So even if they get the same HLA match within a family versus with a stranger, the chances of graft acceptance are closer within a family because there's going to be less differences in all those minor HLA antigens. I don't know the rules for each kind of organ, how close the transplant has to be. This is a theoretical, this is theory that can be done in laboratory animals, but it's not currently, people are not currently transplanting hematopoietic stem cells and solid organs and thymus in humans that I know of.
It's possible to transplant like a small amount of thymic tissue into, I mean, theoretically, to educate.
It's easy to do in laboratory animals.
You don't need a full functioning thymus?
No. It seems to sort of get hooked up to the circulatory system and do what it's supposed to do spontaneously. In fact, if you take, so we know that there are genes that are involved in TCR and BCR gene rearrangement. In the mouse, they're called RAG. So RAG knockout mice have no T cells and no B cells. There's essentially no adaptive immune system. Not only can you do this transplantation where you put the thymus between another mouse gene, but you can actually do this with human tissue. You can put a piece of a human thymus, human hematopoietic stem cell, into those mice, and they develop a human adaptive immune response that is tolerant to the mouse antigens through the mouse thymus and the human antigens through the human thymus. Okay, just a few more minutes about hematopoietic stem cell transplantation. So obviously, in order to get a new immune system in a transplant recipient, you need to transplant cells that can proliferate. So just having mature B and T cells is not a whole immune system. So to reestablish an immune system in a recipient who gets a lethal dose of radiation or chemotherapy to eliminate tumor, you need to transplant hematopoietic stem cells that are capable of reproduction. Those result in a self-sustaining population of hematopoietic stem cells in the recipient, and then can lead to all the circulating progenitors, the common myeloid progenitor, the common lymphoid progenitor. So the ways to make it successful are to myeloablate the recipient, so either by radiation or chemotherapy, the host hematopoietic cells are eliminated. HLA matching also helps. And then during the time that the recipient has no mature immune system, they have to be supported with blood transfusions and anti-infectious methods to give the new immune system time to differentiate mature lymphocytes. And we actually already talked about this, that if you do a hematopoietic stem transplantation and have some slight antigenic mismatches, that the new immune system can help eliminate lymphoma with the graft versus lymphoma or leukemia response. Any more questions about transplantation? It's kind of a mind-stretching subject, even if it's not against your beliefs. And I'm happy to keep chatting about it with anybody. Otherwise, you can take a 10-minute break and we'll start again at 2.
This is